Hi guys, in this video we will be explaining about glycolysis, Krebs cycle and electron transport chain. So first of all, let's talk about glycolysis. Glycolysis is the metabolic pathway that converts glucose into pyruvate. Glycolysis takes place in cytoplasm, and there are 10 enzymes involved in breaking down the sugar in this process. Now, I will be explaining the process of glycolysis. So let's go to step 1 first. In this step, the enzyme called hexokinase adds a phosphate group to glucose in a cell cytoplasm. In this process, a phosphate group from ATP is transferred to glucose, producing this glucose 6-phosphate or G6P. So in this process, one molecule of ATP is consumed. Next, step number 2. In this step, the enzyme phosphoglucomutase isomerizes G6P into its isomer, which is fructose 6-phosphate. Isomers have the same molecular formula as each other but different atomic arrangements. Next, on step 3, the enzyme called phosphofructokinase uses another ATP molecules to transfer a phosphate group in order to form this fructose 1,6-diphosphate. So far, 2 ATP molecules have been used in this process. Next, in the step number 4, an enzyme called aldolase splits up this fructose 1,6-diphosphate into 2 different molecules, which is a ketone and an aldehyde. These 2 molecules are isomers of each other. Next, on the step number 5, the enzyme called triose phosphate isomerase rapidly converts these molecules, DHAP, into these molecules, GAP. These molecules is the substrate needed for the next step of glycolysis. Next, in step 6, the enzyme called glyceraldehyde 3-phosphate dehydrogenase or GAPDH serves 2 functions in this reaction. First, it dehydrogenates GAP by transferring one of its hydrogen molecules to the oxidizing agent NAD to form NADH. Next, GAPDH adds a phosphate from the cytosol to form this 1,3-diphosphoglyceric acid. Both molecules of GAP produced in the previous step undergo this process of dehydrogenation and phosphorylation. Next, for step number 7, the enzyme called phosphoglycerokinase transfers a phosphate from this molecule to a molecule of ADP to form an ATP. This happens to each molecule of this 1,3-diphosphoglyceric acid. This reaction yields 2 of this 3-phosphoglyceric acid and 2 ATP molecules. Next, for step number 8, the enzyme called phosphoglyceromutase relocates the P of these molecules from the third to the second carbon to form 2 of 2 phosphoglyceric acid molecules. Next, for step 9, the enzyme called enolase removes this molecule of water from this molecule to form this PEP or phosphoenol pyruvic acid. This happens for each molecule of 2-PGA from step 8. For the final step of this process, the enzyme called pyruvate kinase transfers a P from this PEP to ADP to form ATP and pyruvic acid. This happens to each molecule of PEP. This reaction yields 2 molecules of pyruvic acid and 2 ATP molecules. For the final results, this process yields 2 pyruvic acid, 2 ATP, and 2 NADH. For results, this process yields 2 pyruvic acid, 2 ATP.
In this video, we will talk about Krebs cycle. Krebs cycle is also known as the tricarboxylic acid TCA cycle. It is a biochemical pathway that is used to generate energy through the oxidation of acetyl-CoA. It is also used for the synthesis of NADH and for the production of amino acid. Krebs cycle takes place in the mitochondria of the eukaryotes and cytosol of prokaryotes. The first step involved is actually a preparatory step which begins with pyruvate. Pyruvate is derived to the glycolysis of glucose, which is 6-carbon compound, and it is split into 2 molecules of pyruvate, which is 3-carbon compound. The next step includes the oxidation of pyruvate into acetyl-CoA by the enzyme pyruvate dehydrogenase complex, and in this reaction, a molecules of carbon dioxide and a molecules of NADH is generated. The acetyl-CoA is a 2-carbon compound. In the next step, the acetyl-CoA combines with oxaloacetate, which is 4-carbon compound, to form citrate. Hence, the resulting molecule is 6-carbon compound. This reaction is catalyzed by the enzyme citrate synthase. In the next step, the citrate is isomerized into isocitrate by the enzyme aconitase. Next, the isocitrate is oxidized into alpha-ketoglutarate by the enzyme isocitrate dehydrogenase. In this reaction, NAD is reduced to NADH and carbon dioxide is generated. Hence, the alpha-ketoglutarate is a 5-carbon compound since 1 molecule of carbon is lost into carbon dioxide. Next, the alpha-ketoglutarate is converted into succinyl-CoA by the enzyme alpha-ketoglutarase dehydrogenase. In this reaction also, a molecule of NAD is reduced to NADH and a molecule of carbon dioxide is released. Hence, the succinyl-CoA is a 4-carbon compound. In the next step, the succinyl-CoA is converted into succinate by the enzyme succinyl-CoA synthase. In this reaction, a molecule of GTP is generated. Next, succinate is converted into fumarate by the enzyme succinate dehydrogenase. In this reaction, a molecule of QH2, which is used for the production of FADH2, is generated. Fumarate is then converted into malate by the enzyme fumarase. In the last step, the malate is converted into oxaloacetate by the enzyme malate dehydrogenase. In this reaction, NAD is reduced to NADH. Now, look at the result of Krebs cycle. Through each cycle of the Krebs cycle, following products are generated: 3 molecules of NADH, 1 molecule of FADH2, 1 molecule of GTP, and 2 molecules of carbon dioxide. Now, since glucose is split into 2 pyruvate compounds for each molecule of glucose, this cycle runs twice. Hence, the product form are 6 NADH, 2 FADH2, 2 GTP, and 4 carbon dioxide. All the NADH and FADH are next to fed to the electron transport chain for the generation of ATP. Now, I will explain how to memorize the Krebs cycle. To memorize the Krebs cycle is to know about the key molecules. There are 8 key molecules that make up the Krebs cycle. In these, citrate is the first molecule. Here is the simple acronym to memorize 8 key molecules. Can I keep selling substance for money, officer? C stand for citrate. I stand for isocitrate. K similarly stand for alpha-ketoglutarate. S stand for succinyl-CoA. And the other S stand for succinate. F stand for fumarate, and we have malate, and we have oxaloacetate. The best way to remember the carbon count is to think about the process starting with 6 carbon citrate. The carbon is dropping off from here until we get 4 carbon of oxaloacetate again. So, acetyl-CoA has 2 molecules of carbon and oxaloacetate has 4 molecules of carbon. They combine 4 plus 2 to form the 6 carbon of citrate. In the next molecule, there is no change occur, just molecules and rearrangement occur, so 6 carbon again. In the next reaction, we remove on carbon again to produce the 5 carbon compound of alpha ketoglutarate. The carbon removed is lost in the form of carbon dioxide. Next, we again drop 1 carbon in the form of carbon dioxide to produce 4 carbon succinyl-CoA. From here and forward, the count remains the same until oxaloacetate, succinate, fumarate, malate, and oxaloacetate all have 4 carbon until oxaloacetate combine with another molecule of acetyl-CoA to form citrate.
Now, I will be explaining the last steps of cellular respiration, which is electron transport chain. This process happens in the mitochondria. So, in this process, the electrons from glycolysis and Krebs make their own way from this mitochondrial matrix to the inner membrane space. And that's why the process of electron transport chain is occurring here in the inner membrane space. So basically, in this process, electron energy is used for pumping proton from the mitochondrial matrix to the intermembrane space to increase the concentration of proton here in the intermembrane space. So the only way for the proton can escape is through a channel and an enzyme, which is this ATP synthase, which uses diffusing proton's kinetic energy to make ATP from ADP and P. So I will be explaining the process in more detail now. First, it is an ADH that starts this process by donating electrons to the complex number 1. Then this powers activate transport as protons are displaced and get pumped from the mitochondrial matrix to the intermembrane space. From complex 1, electrons flow to ubiquinone, and the electrons are transported by this ubiquinone to complex number 3. The function of complex number 3 is the same as complex number 1, which is as a proton pump by using electron energy. Meanwhile, complex 2 is for FADH2, which donates electron and passes the electron to ubiquinone and later on pass them to complex number 3. From complex 3, the electrons proceed to another mobile carrier, which is cytochrome C, which donates the electrons to complex 4. Complex 4 has the same function as complex 1 and 3, which is as a proton pump. After complex 4 electrons flow, electronegative is keep on pulling the electrons down the chain. And during this process, oxygen grabs protons from the matrix to form H2O or water. Now, all of the protons are trapped in the intermembrane space and cannot go anywhere. But same as the other particles, these protons are attracted to go from a high concentrated place to a lower concentrated place. So they finally diffuse to the mitochondrial matrix through this ATP synthase channel. The matrix site of ATP synthase has binding sites for ADP and P, which come in and bind, which catalyzes chemical bond formation. So finally, ATP is formed by ADP and P. and ADH that enters the electron transport chain at the beginning, yielding 3 ATP per NADH, while FADH2 that enters the ubiquinone, producing only 2 ATP per FADH2. So that's all for our explanation about glycolysis, Krebs cycle, and electron transport chain. Thank you for watching. Krebs cycle and electron transport chain. Thank you for watching.